So the next component of the phospholipid bilayer or membrane is termed as cholesterol. So cholesterol, again, they are lipids molecule. So majority of them, you can see that you get it in the animal cells. Plant, they don't have cholesterol. So plant, they don't use cholesterol in their membrane. They use a different kinds of the sterol. Okay, they use a different kind of sterol, not cholesterol. Yeah, be careful. Okay, we always claim plant they don't have cholesterol. So they use a different kind of cholesterol. It's named as sterol. Okay, so biosynthesis in animal cells because essential structural component of all animal cells membrane. So in terms of cholesterol, they are amphiphilic. So amphiphilic, amphibians means like water, also like no water, right? So what does it mean amphiphilics? Basically, they're similar to the phospholipid bilayer. They do have the hydrophilic portions and hydrophobic region, but don't claim it's a head or tail. Are you clear? So you just say that they have hydrophilics and hydrophobic region. So what does it mean amphiphilics? Means that they have a hydrophilic portion of cholesterol. Can you see that? OH group. I always say the OH group. They are hydrophilics. They also have hydrophobic regions. So this is the hydrophobic region. But because the hydrophobic region is larger compared to the hydrophilic regions, so cholesterol actually insoluble in water. Are you clear? Okay. Only small part actually like water. The majority don't like. So they follow majority. So in this case, cholesterol molecule is water insoluble, but they soluble in lipids. Okay, and cholesterol, even though they have a small part like water, but they still can diffuse across the phospholipid bilayer. Are you clear? Okay, so the hydrophilic regions of this cholesterol bonds to the hydrophilic heads, and hydrophobics definitely they work together with the hydrophobic tail. So you look at the structure here. Can you see that? The small dot here indicates the OH group. So this OH group actually can interact with the water molecule or interact with the head of the phospholipids because they are hydrophilics. Can you see that? Hydrophilics. So, but the ring structure of the cholesterol, the remaining structure of cholesterol, they are hydrophobic. They don't like water, but they like the lipids. So they can interact with the fatty acid tail by hydrophobic interactions again. So cholesterol, this is very, very important. They are bidirectional regulator. Students always confused with this bidirectional regulator. So bidirectional basically means that at different temperature here in this context, at different temperature, they have different role. Okay, now imagine, sir, at high temperature, membrane will be too fluid. Correct or not? High temperature. So means that what? In high temperature means that kinetic energy, okay, will be high. So means that the membrane tend to be too fluid. Try to imagine vibrates. So if we don't have something to help them, they're too fluid until the membrane is too fragile and the membrane will lyse. So the cell will die. Are you clear? Okay. So what will happen here is, if between the phospholipid molecule, you have something to interact with, can I see that? Something to tie them together, they won't be able to break apart. So the membrane integrity can be maintained. And this only happens when high temperature, which indicates that kinetic energy is high. Are you clear? So kinetic energy is high means that the molecule will vibrate too vigorously until the membrane will spoil and damage. So in order to prevent this, we do have the cholesterol molecule in between of them. Can you see that cholesterol molecule in between of them? So that this cholesterol molecule will going to hold the membrane, so the membrane won't be too fluid. Are you clear? Okay. But at the same time, when the temperature is low, okay, so means that when temperature is low, lower temperature indicate what? Kinetic energy is low. When kinetic energy is low, means that the membrane is less fluid, and it's bad. Too viscous is very, very bad. Why? diffusion can't take place, right? Because the vibrations of the molecule, actually help us to have the diffusion. But now, without the vibration or very little vibration, what will happen here is diffusion can't take place in the high rate. So what we're going to do here is something like you pack the book, one book, second book, okay? You can pack them nicely, your book, right? But if, let's say, between the book, you put a pencil there, you can't pack them nicely, right? Correct or not? Okay, so the pencil here actually is something like the cholesterol. So when you have the cholesterol in between, can I see that? In between the phospholipid molecule, so it tends to make the membrane increase in fluidity. So if the question asks you about the cholesterol, what is the function of the cholesterol? Okay, we always go for when low temperature, they maintains the or regulates the membrane fluidity or increase the membrane fluidity. Are you clear? Okay. But if the question asks you for 2 marks, then you add the second one and high temperature. So when you talk about the cholesterol role, temperature is very important, because if you didn't tell me the temperature, then I do not know whether you understand the concept or not, because cholesterol only regulates or maintains the membrane fluidity when the temperature is low, but it maintains the membrane integrity when the temperature is high. So this is very, very important. When you explain in your answer, you need to include, right now, you need to include the temperature, at which temperature, okay? So you don't say maintain or regulates the membrane fluidity, full stop. When? They only regulates or maintain the membrane fluidity when the temperature is low, okay?